Assalamualaikum and salam sejahtera, Madam Moderator, AP Dr. Wang Seok, our esteemed invited speaker of the day, Professor Dr. David Perera, and distinguished participants. On behalf of the Organizing Committee and MMP Department, it gives me great pleasure to welcome everyone to today's webinar titled COVID-19 Emergence of Variants of Concerns. For your information, this webinar is the first webinar of MMP Webinar Series, whereby there will be more webinars to be held in the coming years, inshallah. Ladies and gentlemen, for the past 20 months or so, we have been grappling with COVID-19 pandemic. As of yesterday, the total number of positive cases in Malaysia is around 1.9 million, with a devastating death toll of 19,163 cases. However, it is very encouraging that of late that we are seeing a decreasing trend in numbers of admission to hospital and ICU admissions. With the current national vaccination coverage rate among the adult population reaching 70%, it seems that we are finally seeing the light at the end of the long dark tunnel. However, the emergence of variant of concern, although not totally unexpected, is very concerning. The CDC has listed a few of VOCs, and these VOCs has been found to be circulating among Malaysian population. Under the collaborative effort between KPT and MOSTI, several institutions have been doing genome sequencing and surveillance in Malaysia. Among these institutions is the UNIMAS Institute of Health and Community Medicine, IHCM. IHCM is doing the genome sequencing and surveillance in Sarawak, and recent data shared by the CPRC have shown that out of 1,000 VOC cases detected in Malaysia, a large number of these cases are actually being reported in Sarawak alone. Thus, we are very privileged today to have the director of IHCM, Professor Dr. David Perera, to share his valuable insights on the COVID-19 VOCs. The emergence of VOCs, with the emergence of VOCs, there are concerns that how this will impact on the pandemic in terms of disease transmission, disease severity, diagnostic capabilities, vaccine efficacy, and public health measures. And I believe Professor Perera's talk today will shed some light on these crucial questions. Ladies and gentlemen, MMP department is heavily involved in both diagnostic and research of COVID-19. Our diagnostic lab receive an average of 20 to 30 suspected cases daily, and most of our lecturers are currently involved with many various COVID-19 related research. As part of an academic institutions, MMP department is very much committed in promoting knowledge advancement, and we hope that today's webinar serve as an information sharing and intellectual discourse platform. On that note, let me welcome everyone again to today's webinar, and I hope today's webinar will be an exciting, enjoyable, enlightening experience for all of us. We look forward to your future participations in our future events. Thank you very much. Madam Moderator, back to you.
Okay, thank you, Dr. Noraziah. It is a great pleasure today to have Professor Dr. David Perera as the speaker. Professor Perera is the director of the Institute of Health and Community Medicine at Universiti Malaysia Sarawak. His research interest is in the genetics, pathogenesis, and molecular epidemiology of clinically important viruses in the region, including SARS-CoV-2, particularly in the island of Borneo. He has published more than 40 scientific journal articles with more than 4,800 citations of his work and H-index of 27. He has also been the recipient of numerous research grants from several local and international funding agencies, including from the National Institute of Health, USA. Professor Perera has extensive international research collaborations and actively supervises a number of local and international PhD and MSc candidates. Institute of Health and Community Medicine, headed by Professor Perera, is a member of the National COVID-19 Genomic Surveillance Consortium under MOH, MOSTI, and MOHI. The role of this consortium is to increase the capacity of genome surveillance via sequencing in the collaborative effort. Today, Professor Perera will share about the latest updates in regard to COVID-19, the emergence of variants of concern. Without further delay, I would like to hand over the floor to our esteemed speaker, Professor Dr. David Perera.
Hi, thank you. Can you hear me? Seok?
Yes, can.
Okay, great. So thanks AP Dr. Wang or Seok. By the way, Seok was a lab member with me, a colleague during our student days. Yeah. Thank you to the department head, Dr. Noraziah, for the invitation to give this webinar. And most of all, thank you to all the participants today for giving up a little bit of your lunch break hour to sit down and hear yet another thing about COVID. So maybe I hope at least that this presentation will be worth your while to give up some lunch, or maybe you can have your lunch while listening to this presentation. I would also like to take this opportunity to give a shout out to my recently reconnected good buddy from our undergraduate days, Mr. Leong, who is tuning in all the way from Melbourne. I see he's logged on. So hi, buddy, and we'll talk more on Saturday. So I'm going to start sharing my slides now. Moderator, can you allow me to share my slides?
Yes, Prof. David. Can you share a slide?
Okay, now I can.
All right.
Okay, can everyone see the slides?
Yes.
All right.
Can you please put into the presentation mode, please? Yeah.
Just about to do that. Okay.
Yeah, thank you.
Right. So today we're gonna talk about COVID-19 and all about variants of concern and also variants of interest, what they are, what they mean to us, how do they impact us, particularly with this current pandemic and this current surge that we are seeing, not only in Sarawak, but in Malaysia as a whole. So this webinar series, as was mentioned by Dr. Noraziah and Seok, is a collaboration between the Department of Microbiology and Parasitology, Faculty of Medicine at UiTM, together with the Research Innovation Department of UNIMAS. So it gives me great pleasure to share with you some of my thoughts on emergence of variants of concern. So let us start with some of the sobering numbers as of yesterday, the 8th of September. As correctly pointed out by AP Dr. Noraziah just now, Malaysia has now recorded almost close to 2 million confirmed cases of COVID, with fast approaching 20,000 deaths. Now, that's not a trivial number, because for a small country like with a population of approximately 33 million, this certainly is something very impactful to the country. As a whole, globally, the world has seen more than 200 over million cases with more than 4.5 million deaths. So certainly the COVID story over the last coming to 2 years now has greatly impacted the entire globe, including here in Malaysia. Now, I took this from Johns Hopkins data center as of yesterday. If you look at the daily confirmed cases 7-day average or per million people, in all the continents, particularly in North America, in Europe, in Asia, in Australia, you see a surge. There's a rising surge of cases. And to a large extent, this is due to the spread of the Delta variant. The only drop that can be seen is in South America, right, at the moment. But everywhere else, cases seems to be increasing. Now, in Malaysia, for a country of our size, the cases per million is quite large indeed. In fact, we are one of the top few in the world with a number of cases per million population. Not something we want to be proud about, but something we should be alarmed about. Now, when we talk about SARS-CoV-2, I thought how I would model the presentation today is take you through a little bit about the classification, characterization, and to help us to understand a little bit more about the virus itself. Because this will help us to appreciate when we talk about mutations and variants of concerns or variants of interest later on, to appreciate what do they mean and how does it impact the role of this virus when it comes to infection. So in terms of SARS-CoV-2 classification, I mean, for most of you, you know, if you look at all these orders and families and subfamilies, it's not something that we want to remember, right? I mean, I don't typically would remember all this, but maybe what we want to take away from here is that for human coronaviruses, generally they come from two genuses, right? You have alpha coronaviruses and the beta coronaviruses. And to date, our common cold, right? There have been 4 coronaviruses, two in the alpha and 2 in the beta, these ones here, that have been associated with the human common cold. And we get the common cold almost every other year. Some of us, almost every year, you get the common cold, right? That in itself suggests that, you know, you probably don't have very much long-term immunity to these coronaviruses from the fact that you can get infected almost every other year. But in recent years, we have seen the emergence of newer human coronaviruses. In 2003, of course, we had the SARS coronavirus, and that was a major outbreak, causing over 800 deaths worldwide. But a series of changes in the virus, predominantly a set of deletions in its genome, sort of like rendered the virus incapable or less pathogenic or to be transmitted. So much so that since then, there have been no reported cases of the SARS coronavirus since. Then we had the MERS coronaviruses that was first detected in camels in Saudi Arabia in 2012, and then associated with a lot of the Hajj participants in Saudi Arabia. And these also caused quite a number of deaths and was very concerning, but never went on to develop into a pandemic status. Then, of course, now we have SARS-CoV-2, which is a cousin of the other two viruses, first discovered in Wuhan in late December 2019, and since has gone on to become a pandemic and cause untold number of deaths and health burden to almost all countries in the world. Now, if we look at the genome and structure organization of this virus, here on the left, you see electron micrograph taken from CDC pictures in the US of the virus that we are talking about. The term coronavirus actually comes from, if you look under the EM, this corona that you see around the virus particle, which is caused by these spikes, right? So it's quite beautiful actually. If you see it, you see, you know, it's like a glowing sun or the corona around the sun. And in a transmission electron micrograph, you can see what you call it, these little spike structures, right? That in this illustration is more aptly demonstrated. So this is the spike protein, these little protrusions here. And then it's an envelope virus with a membrane protein. You have a nucleocapsid protein that's associated with the RNA genome of this virus. This virus is an RNA virus. And you have the envelope protein with a lipid membrane, right? Encased with a lipid membrane here. Now, if we compare the genome organization of COVID with MERS and SARS, the original SARS coronavirus, the cousins of SARS-CoV-2, we see that the genome arrangement is similar but not the same. The virus is approximately has a genome of 29.9 kilobase pairs. That's about 29,000 nucleotide elements in the genome. And it's differentiated into segments that are non-structural in nature. That means they don't code for the structure of the virus. And then you have the structural components coded for in the virus genome. So you have the spike, the membrane protein, the envelope protein, the nucleocapsid, and so forth. Right, so you see there are subtle differences in the genome organization, right? The sizes are not exactly the same for COVID, MERS, and SARS, the original SARS. And if you just look at the spike protein itself, and we are concerned about this because this is what we predominantly discuss in association with variants of concerns or variants of interest. You see that COVID-19 and the original SARS have somewhat similar designation, but a little bit more different with the MERS coronavirus spike protein. Now, generally, the spike protein is broken up into two parts. You have the S1 subunit and the S2 subunit. The thing to note with the S1 subunit is the receptor binding domain, or RBD. This is the part of the spike protein that interacts with the receptor on the host, or this is the one that sticks to the human cell. And that's this part, the knob part of this spike protein. The stem part here that sticks into the virus membrane is the S2 subunit. Okay, and now I can't, can we remove this annotation thing? Yeah, okay. Now, when we talk about SARS-CoV-2 and VOCs or VOI, we need to talk about mutation, because these VOCs, these variants, they occur because of small changes that happen in the replication of the virus. So when viruses replicate, they change a little bit during the replication process. And this is very normal for a virus. And the reason a virus does this, because it needs these changes to adapt or to changes in its environment to allow it to continue to thrive. So these little changes, we call them mutations. And if enough of these mutations occur that make it a bit different from its original parent virus, then we call it a variant. So for RNA viruses, which is what SARS-CoV-2 is, these changes occur because of faulty RNA-dependent RNA polymerase. This is a protein which is the copying machine of the virus. So it's coded for by the virus genome. And this protein is responsible for copying new progenies of the genome when the virus replicates. And sometimes it makes errors. And this is typical of the majority of RNA viruses. So how much faultiness, so to speak, does the SARS-CoV-2 RDRP or RNA polymerase have? If you look at other RNA viruses, if you take measles, for example, it has a very low mutational rate. The number of changes in the nucleotides, the elements in a particular site on the genome each year, it's very low. So measles is quite stable. And because of that, we can see that vaccination against measles, possibly the reason for it, gives you very long-term protection because it doesn't change very much. HIV, by contrast, has a much higher mutational rate. And so it changes quite often to evade challenges to the virus trying to be dominant in its environment. If you take influenza A, which causes the flu, it has quite a high mutational rate. And this is the reason why in temperate countries like in North America, for example, a continuous ongoing surveillance of the influenza or the flu virus variants or genotypes are ongoing to determine what is the best fit for the vaccine type that needs to be given for your flu shot on an annual basis, right? So you need to continue to monitor whether there are changes to the genotype or the variant in a particular flu season because of the fact that the flu virus can mutate much more. If you look at the original SARS-CoV-2, it has a relatively stable, there's quite a range for it, but on the lower end of it, as with SARS-CoV-2, the mutation is somewhat slower compared to that of the flu. So while SARS-CoV-2 is not a fast evolving virus, it still has a significant enough mutational rate to give it some changes and therefore creating new variants. But we also have to take into account that when a virus achieves a pandemic status like SARS-CoV-2, then the petri dish, so to speak, becomes much bigger because now you have the entire global population behaving like a petri dish for the virus, and therefore the virus has more opportunities to mutate and to churn out variants, even though traditionally it would have a rather low or stable mutational rate. Now, in terms of nomenclature, how do we go about naming all these SARS-CoV-2 genetic lineages or variants, right? So scientists, they get together, and from pretty much the very beginning of the pandemic, 3 separate nomenclature systems developed at GuySide, NextStrain, and Pango have been used as a means to track and name these different variants. Recently, to assist with public discussions of variants, WHO recommended using more friendly nomenclature designations using the Greek alphabet, i.e. alpha, beta, gamma, delta, and so forth. This was to avoid any stigmatizing of the naming conventions used earlier, whereby we used to call, for example, VOCs, the South African VOC or the UK strain or the Indian strain. So to avoid this stigmatization, WHO has recommended the use of this Greek alphabet as a means for public forum discussion, right? However, in a scientific environment, most researchers predominantly use the Pango nomenclature systems when referring or in discussion in a scientific context to genetic lineages. So the general naming conventions for the Pango system consists of an alphabetical prefix and a numerical prefix. So each dot, when you see now, I think most of us are very familiar with a B dot something and something. The dot means descendant of in the Pango nomenclature system. And is applied when one ancestor can be clearly identified, meaning when a mutation occurs in the genome and it accumulates enough to be significantly different from its parent, then you put a dot and give it another numerical suffix. So for example, lineage B.11.7, which is the alpha VOC, is the 7th named descendant of the lineage B.1.1. And C.1 is the first named descendant of the lineage C. Now, the Pango system limits it to 3 descendant categories only here. So if there's a 4th, then they switch to a different alphabetical prefix, right? So in the case of Delta now, there are subvariants of the Delta variant, which is now called AY, right? So you can see a string of AY if you look into the Pango website. So what are variants of concerns or variants of interest? So this is the WHO working definition of variants of concerns. Now, a variant concern must meet a definition of a VOI. So it must have first been a VOI and must be demonstrated to be associated with one or more of these things, right? Increased transmissibility, meaning very infectious. Increased virulence, causes more severe disease. And a decrease in effectiveness of public health measures, meaning it affects your diagnostic measures. Maybe the diagnostic tools that we are using now picks up more, what you call it, false negative results. So the tests are failing. Or if our vaccines become ineffective, or therapeutics such as certain monoclonal antibodies that are being used as treatments for COVID-19 now has a decreased efficacy with regards to treatment. So when this have a high degree of global public health significance, then the WHO may consider naming these variants as VOCs. Now, a prelude to VOCs are, of course, variants of interest. This is to highlight certain variants that have the potential to become VOCs in the future. And so a red flag is put up beside these variants to help the researchers or the governing bodies to be aware and keep a close watch on these VOIs in the event they turn to become a VOC in the future. So what are VOIs? These have very small genetic changes that also affect characteristics such as transmissibility, severity, immune escape, diagnostics and therapeutics, but have not yet reached the scale of global public health significance like a VOCs, but have the potential to do so. So, and these must have been identified to cause significant community transmissions, because VOIs can independently evolve in different community settings, like in different countries, for example, or different geographical locations, for example. We will see later in these presentations, there's an example for this in which in Sarawak, we have seen a variant that possibly could be called a VOI. So what are the currently designated VOCs that have been listed by the WHO? You have the alpha using the friendly WHO Greek label, which in the Pango lineage is called B.1.1.7. First documented sequence was from the United Kingdom in September 2020. This has now been shown through various publications and various studies to have an approximately 50% increase in transmission rates. In fact, in the UK, when the alpha variant hit, it practically swept through the entire country. It has shown to have a potential increase in terms of disease severity. It did not appear to have any impact on the currently used monoclonal antibody treatments. As I know of, there are at least 4 different monoclonal antibody treatments for COVID-19, and for alpha, it doesn't seem to have any impact on these. And it seemed to have minimal impact on neutralization by convalescence and post-vaccination sera. This means that people who have recovered from infection, whether it was from alpha or from any of the earlier variants that were circulating, seem to have protection against alpha. And people who have received vaccination seem to get protection from that vaccination against the alpha variant. And then we had the second VOC, which was the beta, that was first seen in South Africa in May 2020. The Pango lineage for this was is B.1.351. It was also shown through various studies to have a 50% increased transmission rate, so highly infectious. It had, when compared against alpha, significantly reduced the susceptibility to some of the monoclonal antibody treatments. So basically, some of these monoclonals now had less efficacy in terms of its treatment ability. And there was reduced neutralization by convalescent and post-vaccination sera, meaning to say that people who had recovered from the disease earlier did not have the protection against, adequate protection against the beta. And this was also so for people who had received 2 doses of their vaccination, right? There was reduced protection. So in a sense, there was some vaccine escape or immune escape for the beta variant. Now, the other VOCs, and there are 4 in total currently designated by the WHO. We have, of course, what's sweeping the globe now, which is the Delta variant, the Pango lineage designation B.1.617.2. And you have further dots now, and because the Pango lineage uses up to only 3 dots numerical prefix, they are now all called AY. And you have a whole range of AYs designated for the Delta. So you have basically sub-lineages of Delta circulating in various places around the world now. So it was first identified in India in October 2020. Various studies have now shown that it has definitely increased transmissibility. It has potential in the reduction in neutralization for some emergency use application of monoclonal antibody treatments, but not all. And it has shown potential reduction in neutralization by post-vaccination sera. Now we will look at a little bit more data on vaccination later on in this presentation. So the Delta has shown that it may potentially have a big impact on treatment and vaccination. And so it is something that we need to be concerned about. And then lastly, you have the gamma or P.1 VOC, first identified in Brazil in November 2020. This particular VOC has been shown to be significantly reduce the susceptibility to some monoclonal antibody treatments, but not all of them. And it has shown to have reduced neutralization capacity when convalescent sera of people who have recovered from the disease and people who have got their 2 doses of vaccination against this particular VOC, this gamma. But do we still need to be concerned about all 4 of them? We'll see in a short while. But before we go to that, now why do we want to talk about these VOCs? Now, when VOCs are talked about, if you look in the press statements, in various studies, even at our MOH level, they talk about certain key mutations in the spike protein of these VOCs. And the reason they do that is because the spike protein is the predominant protein that interacts with the human host cells during infection. I may have missed, okay, I'm going to go back to this now first. Now, if you look at cell entry of the SARS-CoV-2, when the SARS-CoV-2 binds to a lung cell, for example, right, it uses its spike protein to attach to what is called the ACE2 receptor on the human host cell. So in this illustration here, you have the ACE2 receptor here, and it interacts with, remember what we talked about earlier, the RBD or the receptor binding domain of the subunit 1 portion of the spike protein. So it interacts here. And at the junction between the S1 subunit and the S2 subunit of the stem of your spike protein, you have another protease that's adjacent to the receptor on the host cell that interacts at that junction. So what happens is, if you can imagine, your spike is like a coiled spring, actually. So this knob region here prevents that spring from being released, right? So it's tightly wound up. And so when the knob that has the recognition potential through its RBD to interact with the human receptor, when that happens, then this protease also recognizes the junction between the spike head or the S1 unit and the S2 unit, that is the stem. When that happens, you have a cleavage. So this knob region gets cleaved away. And when that happens, you have the fusion machinery. So basically, the coiled spring is unleashed. And so fusion between the virus and the host cell happens. So you get this bridge, this fusion bridge. And this fusion bridge will draw the SARS particle close to the cell membrane, the host cell membrane, in this case, the lung cell, and fusion of the two will happen. And when this happens, this allows the virus material, the nucleocapsid or the N protein and the virus genome to insert itself into the lung cell. And then various information coded for in the virus genome, the non-structural portions of it, as we had seen earlier, will now instruct the machinery of the lung cell to make more of the progeny viruses. Now we'll go back to this. So how does these mutations on the spike that are on the VOCs, the different VOCs, affect this process? So I've accumulated them here in this diagram, and basically they interfere in two parts of this process. In the binding process of the spike to the human receptor, the ACE2 receptor, and also to the fusion process through the interaction of the cleavage at the junction between the S1 and S2 subunit of the spike. So certain key mutations like the N501 present in the alpha, beta, and gamma VOC, and the K417N or T change in the beta and gamma actually increase receptor binding. So if you have increased receptor binding, then you have more efficient infection of the virus to the lung cell, right? So how to read this? This means N501Y means that this particular amino acid, which is the building blocks of the protein, has now been changed from an N to a Y, a different amino acid. The N was in the original Wuhan strain, and now in, oops, sorry, I clicked it. In alpha, beta, and gamma, this N has now become Y. And similarly, all these changes on the left here is the original Wuhan amino acid, and all the ones alphabets on the right here are the changes that occur in the various VOCs. So these two key mutations here that have evolved on the virus spike protein allows the virus to have more efficient binding to the host cells. And then you have these mutations that affect, that are generally on the periphery of the binding of the RBD on the spike to the ACE2 receptor. And these sort of like put up a wall that prevents neutralizing antibodies to interact with the virus particle. So you have three key mutations here, the E484, which is present in alpha, beta, and gamma VOCs. And then you have these two that are present in delta, 478 and 452. And together, these mutations confer the ability of the SARS variant to evade neutralizing, or in other words, to reduce the neutralizing capacity of either people who have recovered from COVID or people who have had 2 doses of the vaccination. So there may be reduced capacity of these people to effectively neutralize the SARS-CoV-2 virus. So you have reduced protection, right? So these are something we would be concerned about when we talk about vaccine protection. Of course, VOCs are not the only things that we should be concerned about. There are also VOIs, or variants of interest. And these are things that we need to keep an eye out for because they have the potential, of course, to become VOCs. And 5 have been identified and designated so far by the WHO. You have ETA here, which is in the Pango lineage B1.525. Its key spike mutation here is the 484. Right, and if you remember from the previous slide, this is the one that's involved in reduced capacity. So this has been shown through various studies that it has the potential to have reduced neutralization by some monoclonal antibody treatments, and also reduce neutralization by both convalescent and post-vaccination sera. And part of the reason for this is potentially because of this key mutation in the spike, which has been shown for the VOCs to affect neutralizing capacity. And then you have IOTA, which is the B1526. This one has, similar to ITA, also has this particular mutation that's related to reduced neutralization, but it also has an additional unique mutation in the spike head protein that increases receptor binding or to the RBD. So again, it has similar attributes to that seen with the ITA strain, right? Which is reduced treatment efficacy of some monoclonal antibody treatment regimes, and also reduced neutralization by convalescent or recovered people, and post-vaccination sera for people who have received the 2 doses. Then you have kappa, which is the B1617.1. These ones have these key mutations here. The first 2 here, of course, as we had seen earlier in the earlier illustrations, are involved with reduced neutralizing capacity against the virus. And this 5681 here, which is involved in furin cleavage, right? That allows better cleavage to occur so that the virus has a greater capacity to fuse and merge with the lung cell. All these, of course, have the effect of the virus having greater transmissibility and potentially immune escape from both recovered COVID patients and also people who are supposed to be protected from by vaccination. So again, the kappa have similar attributes to both ITA and IOTA. And then you have lambda. Increasingly, there has been some concern about this, and recently reported even in our news media here in Malaysia. So it has a similar mutation here, although with a slightly different amino acid change, but at the same position to that of kappa and other VOCs and even VOIs, that increases its infectivity. But it additionally has a 7 amino acid deletion also within the spike domain on the left end of the spike domain, or we call it the N-terminal domain, that allows it to evade neutralization, either from people who have recovered from COVID-19 or for people who have been fully vaccinated. And so this is something to be concerned about. And then you have the latest one, which is Mu. I like the name Mu, it's nice to say it, Mu. So Mu is B.1621. It has these key mutations that target both the important interactive points with the spike, which is the binding part with the RBD domain to the receptor, and also to the fusion cleavage, which is the P681H here. So it has been shown to have increased transmission, reduces the efficacy of some monoclonal antibody treatments, and also impacts the neutralizing capacity of people who have recovered, convalescents, or people who have been fully vaccinated. So if we look at the global prevalence of VOCs, and this is taken from the GSAID data monitoring site, and these graphs show you the relative variant genome frequency If you look at alpha, so these are all the continental regions, South America, Europe, Oceania, and so forth. Alpha peaked sometime in January to April of this year, and globally it has shown a decline. To a couple of days back, which is here, you are hardly seeing very much alpha. Of course, there's a caveat to all this. These are only sequences that have been made available to GISAID, but GISAID has close to 1 million sequences already deposited globally from many countries around the world. So should we really be concerned about alpha? Probably not. It's declined and probably being phased out. What about beta? Beta never really took off like alpha, right? For Africa, you had a very high prevalence of it for quite some time, but over the last couple of months, there's been a very steep decline in the detection of data, and it never really took off anywhere else, as we shall see with our Malaysian data also later. We did see some beta, but it never really took off on the scale, at least of alpha or delta. What about gamma? Gamma also never really took off. You have it in South America, which had the highest prevalence probably around April, May, but has also now seen a steady decline. For the rest of the world, it never really took off, and everything has been declining in terms of the prevalence numbers for gamma. But what are the different story we have for delta? If you look at delta, it's surging in almost every continent globally. And while it appears to be plateauing in some continents, it's very much still in the exponential phase of its increase. So as you can see from here, there's steep increases over the last few months of delta in every continent on the globe. So I guess it would be safe to say that at this present moment in time, the VOC that we should be most concerned about would be delta. What about the VOIs, particularly this Lambda and Mu that has been making the news recently? Should we be concerned about them?
Sorry,
now,
Prof. David.
Lambda, yes?
It's almost 1 o'clock, it's 12:55 now.
Okay, I will speed it up.
Okay.
I have 2 more slides anyway. So for Lambda, the earliest documented samples was in Peru. It's also showing a steady decline. And for Mu, earliest recorded sample in Colombia, January, also showing a steady decline. So at this present time, there's nothing to indicate that we should be overly concerned about these two VOIs. Now, how much has ASEAN contributed to genome sequencing? You have the numbers here. As of yesterday, Malaysia has contributed just over 2,000 sequences. We are number 5 in ASEAN. These are the percent of sequences, 0.1% of all total number of cases reported in Malaysia. So it's a good start. There's a consortium like Dr. Noraziah was mentioning. So hopefully we will have a sharp increase in the number of sequences contributed by Malaysia soon. Now, of these 2,000, what have we seen? This is the distribution by month from January 2020 to August 2021. These are all the different types of variants that we have seen. Where VOCs are concerned, the ones boxed in red here, this is beta, these ones here in this like yellowish color. And of late, what we have detected are predominantly almost all delta. By distribution of state, most of the data comes from Sarawak. You also have a significant number coming for KL, but there's a bunch of data, about 800 sequences here, where the state designation has not been included associated with the sequence. So we are not sure where these come from, but very likely a lot of them are from the Greater Klang Valley area. These are the contributing labs that have generated sequences. 7 of these are now in the consortium that Dr. Noraziah was talking about, and maybe more may join in the future. Now, this is a little bit about Sarawak per se. I wanted to show this because we see, since we started tracking by genome sequencing, of late, almost all the sequences we see are of the Delta variant, which is this red thing here, right? But in the past, we have seen this yellow one, which is what we call the Pasai, which in the Pango lineage is AU.2. Now, these ones have certain key mutations that potentially could attribute it to be a VOI, and may explain why we saw such a high transmissibility rate for this particular variant and a high spread of this variant before the onset of Delta. When Delta came, it swept everything away. Now, the last part of this talk, everyone's concerned about vaccines and do they protect us against VOC. I apologize for this coming, our talk running a bit late, but if you will give me another 5 to 10 minutes, we will run through about vaccines and how they protect us against VOC. Now, do vaccines really protect us? I take one case study here, a recent one, the Israeli studies done on vaccinated healthcare workers. So they did about 1,400, close to 1,500 healthcare workers who had had their Pfizer 2-dose vaccinations. Of these, 39 had breakthrough infections, meaning they had an infection after receiving their 2-dose of Pfizer. And what they found was that when they measured the neutralizing antibody titers, these are the antibodies that are responsible for sort of inverted commas killing or neutralizing the virus, they showed that higher neutralizing antibody titers, when you have more of it, you have a lower risk of infectivity. They also showed that most of these breakthrough infections were mild or asymptomatic in terms of symptoms, and they showed that predominantly when they had a breakthrough here, the alpha variant, VOC, was the culprit. So when they measured a portion of the virus breakthrough group, they found a mean neutralizing titer of this amount. And when converted to a surrogate virus neutralizing test, now then the neutralizing assay is the gold standard for looking at neutralizing antibody, measuring neutralizing antibody protection. And so that equates to about 60% protection. But in the healthy group, they had about a 78% neutralizing antibody protection. This was protection before they got infected. Meaning to say, if you had this amount of protection, you would have been protected against infection or at least symptomatic disease. But if you had below this, which is in the case of the vaccine breakthrough, they had only 60%, then they were at a higher risk of getting infected, even though they were protected by 2 doses. And what you call it, symptomatic disease. But in this case, it was very mild only. So how well do SARS vaccines protect? We look at the Hong Kong study, a recent Hong Kong study published in Lancet Microbes. They look at 93 participants. This is their preliminary study of their 1-month data. So 63 were given Pfizer or had Pfizer 2-dose, and then 30 had the Sinovac vaccine. And if you see from here, after second dose, a lot of the using the surrogate virus neutralizing test, almost all the Pfizer group had an 80% protection and above. But there was a wide range for the CoronaVac or the Sinovac recipients, from as low as about 30% to as high as 80%. So a wide range of protection efficacy. And when you compare this to the gold standard plaque reduction neutralizing assay, you see a similar trend, right? So in fact, it's a bit lower for CoronaVac, not achieving very high titers, but consistently high titers for the Pfizer recipients. So when they measured the mean neutralizing titer, it was 269 for Pfizer, Sinovac 27. So what does this mean? The authors conclude that the study translates into substantial differences in vaccine effectiveness. Now, should we be concerned in Malaysia because we use these two vaccines? So there's hope yet. When we talk about boosters, let's look at the Chinese study. So in this study that is pre-published in MedRisk, meaning it's not yet peer-reviewed, but they wanted to get the data out to the public, 303 Sinovac participants. So in this study design, the first dose and second dose of the vaccine were given 28 days apart. That's important to note because in Malaysia, we did 21 days. And the third dose was, or booster dose, was given 6 months after the second dose. Now, from the profile, neutralizing profile here, we will see after the second dose, you get a rise in neutralizing protectiveness that wanes, declines over time. It declines over time that approximately 6 months after that, the levels of protection has gone quite low. But when boosted with the third dose, 6 months after the second dose, that protection level increases significantly. In fact, they found a 7-fold increase in the neutralizing titer after the second dose. And this remains so, at least for the study, 1 month after the administration of that third booster dose. So what does it mean? It means neutralizing antibody titers, although it declines substantially after 2 doses, when given a booster after 6 months, this induces a very robust immune response that gives a much higher fold increase in neutralizing protection. And lastly, an Israeli study with the Pfizer vaccine. This was also pre-released in MedRisk, so not peer-reviewed yet. In this study group, they had over 1 million older folks because their booster only given to older folks first. So in this study design, the third booster of the Pfizer was given 6 months after the second dose. And so here they show, in this case, this transient bias is, what's not shown here is the data for the 2 doses. So there is a decline after 6 months, but not to a low level as seen with CoronaVac, but there is a steady decline also. So when given a booster 6 months after the second dose, you see after day 12, and this is typical of how your immune system responds after being given a booster. It takes some time before your antibody levels go up. So after day 12, when measured, they see a 5-fold increase in neutralizing capacity. And this 5-fold was protective against confirmed infection, actually. And they showed that it also corresponded to a greater than 10-fold decrease in relative risk of severe illness. So what does all this mean? This means that the third dose vaccine can both reduce transmission and decrease severe impacts of severe disease, right? And this is potentially important in curtailing the Delta variant resurgence. So points to ponder, the last bit. So what can we take back from this? We definitely need National Genomic Surveillance Program, it's important to know what's circulating. Why do we need to do that? Because we need to know when we have vaccine breakthrough cases, and we are already seeing this, what are the variants that are associated with these cases in order for us to respond or to monitor to the vaccine efficacy. We need to know what variants they are. We need to know what vaccine types have been given to these particular cases that have breakthroughs. We need to have serological studies to determine the neutralizing capacity post-vaccination, because these kinds of data are important to provide informed decisions on the need for the potential third booster and when it should be administered if needed in our population. Here in UNIMAS, in our group, together with the state government, we are being funded to do one such study. And of course, we need public health measures to continue to prevent or limit virus transmission, because the last thing we want is for newer variants to emerge that can totally evade protection from our vaccination. So with that, thank you for listening. Sorry for going a little bit ahead of time, and have a fantastic day ahead. Thank you. Back to you, Madam Moderator.
Thank you very much, Prof. David. It was indeed a very interesting, insightful to the COVID variant. And indeed, it's a very interesting topic that we all want to listen to, and it's with the number of the participants we have, it's almost 300 people listening to this topic during the lunch hour, shows that how much we need to know more about this and confirm about this condition. There are several questions actually lined up in the chat box. I will try to quickly go through them and we see which one can answer. I'll start with the first one from Leon. Says, hi, Prof. David, what is your take on new variant? I think that was answered. And is it now classified as VOI or VOC?
I think that would have been answered already, right? Those,
yes.
The ones listed in the presentation are all the ones that have been designated so far by WHO.
There's a question from Sir Joseph Ramanayar. Prof. David, your view on teenagers and their immune against the virus, is vaccination for these young teens really necessary?
I'm a proponent for it. I think the safety data from Pfizer, which is the only vaccine that has been approved for adolescents 12 to 17 in our country and elsewhere, there's already safety data that's out there, and that's why the FDA has approved and the vaccine for 12 to 17. So it's already improved by our own NPRA, and there's no reason for us not to do it, because we already can look at data and see the impact of it from countries that have already started this long before us. We have Singapore, we have the US, who have already been vaccinating their adolescents. I think it's something that needs to be done fast and needs to be done soon, and good for Sarawak that we have started today actually with our adolescents. Now, it's concerning, and why I'm saying this is because if you look at the US now, they have a major surge because of Delta, and predominantly because almost half the adult population are unvaccinated for one reason or another. So that lot is being impacted with this Delta surge. But in parallel, they're also seeing a major surge of hospitalizations of adolescents. And this is correlated to the opening of a lot of the schools. So obviously, you know, even though adolescents are at lower risk of developing severe disease, but when your numbers are high, then even though you have a small percentage, in terms of real numbers, that can be a big number. And if you are a family, even one kid being sick enough to be hospitalized and God forbid being in ICU is too large a number even. So please, there's no reason not to allow our kids to get vaccinated. So let's just do it.
Thank you for that. There's a question is asking, is there a need to test for SARS-CoV-2 plus flu A, flu B in Malaysia? Would it be
I think MOH has already an ongoing SARI, which is what you call it, a respiratory screening that's done, and I think that will continue to go. So flu will be taken care of from that. But with SARS-CoV-2, I think everyone is testing now, right? If you have any flu-like symptoms or cold symptoms, the first thing your doctor is going to tell you is get tested for SARS. So I think it's already being done. So it's not a question of whether we need to do it. It's already being done.
Yes. There's a question, what is your view on PCR genotyping variant kit to identify VOC and VOI based on the target mutation genes such as L452R and P681R, etc., compared to the use the WGS to identify this VOC and VOI?
Okay, my view on this is that while it's useful in a sense that it's quick, right? I think we have to use it with caution. If we use it as a pre-screen to inform us of a possible VOC, VOI, followed by a confirmation of full genomic sequencing, then that's fine. But to use it on its own as a means of determining whether a sample virus is a VOC or VOI, that's a bit dangerous. For the simple reason that the spike itself is not the only indication of a variant. The variant calling takes note of all changes in the virus, which includes other regions of the virus also that these tests do not look at. And also, if you have a new variant coming in that happens to have, and we have seen how many of these VOCs, VOI share similar mutations, if they also happen to evolve and pick up these mutations, then you have the danger of wrongly calling something a VOC when it could be something new altogether.
Thank you. There's a question on the take on the Ministry of Health announcing that Malaysia working on COVID vaccine produced by Malaysia. Mr. Leong wants to know your opinion on the development of vaccine.
I don't know much about the status of that other than hearing some statements that Malaysia intends to do this with the WHO. All I can say is I wish them well. I mean, they won't be the first. Other countries like Taiwan, Vietnam have also started developing their own in-house vaccine types. So I think we wish our own MOH well, and maybe moving forward, increasingly with the risk of these kinds of things re-happening. I mean, in the last 20 years, we have seen 3 different coronaviruses affecting humans. Maybe that would be a good move moving forward.
Thank you. There's a question from Prof. Edmond asking whether MERS, SARS, and the SARS-CoV were just as infectious and dangerous as SARS-CoV-2 or COVID-19. Why is the manifestation of COVID-19 so drastically serious and out of control?
I think there's no studies to directly compare the 3 viruses in terms of its potential or rate of transmissibility. But obviously, looking at MERS and SARS-1 and noting that it never achieved the levels of transmission seen with SARS-CoV-2, I think it would be safe to say that SARS-CoV-2 certainly is more transmissible, certainly more infectious, but the modes of these transmission levels still need to be elucidated through various studies that actually compares the three.
So there's another question asking whether there have been a monitoring co-infection cases, especially viral co-infection.
I don't think there's a specific agenda to look at that. But having said that, certain platforms that are being used for the genome sequencing, for example, these days, allow you to answer that question simultaneously also. So while there's no standardized or fixed agenda at this time that's actually looking at that question, the fact that we are doing genomic surveillance now with the platforms that we are using will coincidentally allow us to answer this question.
I have a question from Shazrina. I think she's a journalist from the Malaysia now. She would like to ask, she would like to know regarding the contrasting epidemic outcomes between Labuan and Sarawak. Both have fully vaccinated more than 60% of total population, while vaccination have significantly reduced severe and death cases, unlike sharp declines in Labuan, hospitalization admission, and ICU cases are surging in Sarawak, with death also rising. In fact, most of the death cases were adults with comorbidity. So she wants to know, to ask two questions regarding that. One, death case involving adults with comorbidity, what is the significant cause? And the other question is, how about the testing measure in Sarawak, since infection rate in the state is still high?
Well, let's tackle the testing one. For the testing, I think the state authorities and state health authorities have put in place a larger testing capacity, and to that extent, that also reflected in the higher daily reporting numbers coming out from Sarawak. They certainly have attempted to boost or have boosted the testing capacity in the state. Now, in terms of the different scenarios between Labuan and Sarawak, well, we can only speculate. I won't be able to comment in terms of the deaths and comorbids because I'm not privy to that information. That would be a question best posted to the Sarawak health authorities, who will be privy to such information. But perhaps we could speculate if we look at the Hong Kong study that I presented just now. The differences between vaccine efficacy of Sinovac and Pfizer. I mean, in the Hong Kong study, they have seen that the Sinovac recipient neutralizing capacity wanes to a level much lower than that of Pfizer after 1 month. Now, if we take that in context with Sarawak, for example, Sarawak had completed its vaccination. Sarawak and Labuan have completed their adult vaccination much faster than the rest of the country. But the difference being in Sarawak, 80% approximately of the recipients were CoronaVac recipients. The rest being Pfizer and a very small amount of AZAC. The opposite is true for Labuan, right? The majority of the recipients in Labuan were Pfizer recipients. So we can only speculate that perhaps one of the reasons could be vaccine efficacy waning over time, because we started first, and so now our neutralizing antibody levels are waning. So that could explain, and taken together with the Israeli data where there is a threshold protection level for reinfection, perhaps we are below that threshold, and that's why we are seeing more people being infected even though we are fully vaccinated. But at the end of the day, while the numbers may seem alarming, the take back should be that with a high percentage of vaccinations, irrespective of the vaccine type that was given, Sarawak has certainly, both Sarawak and Labuan, have certainly seen, compared to the rest of the country, a reduction in severe cases, right? So category 3, 4, and 5, that's very low. So that's one of the benefits of being fully vaccinated. And we all know that vaccines don't protect you against infection, but protects you against severe disease.
That brings us to another question. The series of questions is regarding the, I think more they want to know on the booster dose rather. There are questions on the efficacy of AstraZeneca vaccination, and if we are gonna be receiving booster dose for Pfizer and Sinovac in Malaysia. And Prof. Raymond wants to know whether are we going to have third booster dose of vaccination. So all this question, I think, revolves about the booster and
well, I think the booster has been in the news. I think everywhere. The US is pushing for it. Israel has started it. The US is going to make a decision on it pretty soon, whether they move ahead with the booster. I think for the above 60, some of them have already got their boosters. Now, the FDA and the WHO stand is that the focus still should be with those who have yet to be vaccinated. With limited resources in terms of vaccine availability, that should be the primary focus. We should be looking at vaccinating all the eligible population that have yet to be vaccinated. So that's the first thing that we should focus on. But after we have done that, right, you need studies that will inform on your population. It's not just a question of a booster being needed or not. Yeah, you can probably determine that from these studies, but you also need data to educate us and give us informed decisions on when to give the booster. It's not like, you know, you see it drop to a certain thing, okay, just give a booster, then you need to prime, you need to see what's the right time to prime your immune system to give you the best results. So that all comes from data from studies, the serology studies that I put up in the points to ponder. So we are doing this study, right? I think there's also a study going on in Peninsular Malaysia. I think has a limited study on this also. But in our study, we will be looking at all three vaccine platforms, right? So you have the Pfizer, you have which is the mRNA, then you have the Sinovac, which we are looking at, which will be the inactivated vaccine, and then we have the AstraZeneca, which will be the adenovirus vector vaccine platform type. So our study looks at all these three platforms, right? It's ongoing. So we hope that the study will be able to generate data that will at least give us a better handle of the antibody protection levels in people who have got these various different vaccines, and what they mean to us as a community, as a nation, in terms of moving ahead with a third booster dose, and when should we have it, and also what type of vaccine should be given.
There is one more question. Does it regarding the, it's more of they want to know your opinion. What is your insight or prediction for the pandemic trend, where how it will end and how it will diminish? There's something that all of us want to know. And there's another one asking whether is it advisable to move on from one vaccination, like from Pfizer after 6 months, to get a second dose of a Sinovac, for example? I think they're talking about the mixed vaccination.
Yeah, okay. So for the pandemic, right, I think as with any other outbreak, viral outbreak, even with the Spanish flu, for example, it will end eventually. How long it ends depends entirely up to us. So it's within our means to dictate how this pandemic will end. How are we going to do that? First, we need to get all the unvaccinated vaccinated. And that includes the adolescents and the kids. So that we leave only a very small proportion of the population unvaccinated for one reason or another. Either they're very young or the anti-vaxxers who just refuse to get vaccinated for one reason or another. So it will end, but when it ends will depend very much on us, right? And then of course you have this issue of the booster, which increasingly looks like something that needs to be given, and probably a reflection of the regime of the vaccine dosage, meaning to say that it may turn out that a 2-dose regime is probably not the optimal regime for most vaccines, at least for the 2-dose ones, but maybe a 3-dose regime would probably give you a prime, a much more optimal protection level. So for the second question, in terms of what we call heterologous vaccinations, that means you get one vaccine type first, followed by a different vaccine type. Now, there's a lot of studies being done, particularly with Moderna in the UK, with AstraZeneca. Thailand is also doing mix and matching. And the initial data coming out from these studies shows that the mix and matching certainly boosts protections to levels higher than homologous vaccination, i.e. with the same vaccine type for the 2 doses. However, the concerns for safety have not fully been addressed yet. So until that has been addressed and we have the safety data for that, then we'll see whether that kind of heterologous vaccine regimes will be something that will be offered to the general public soon.
There's one last question from the chat asking if they're looking at the current majority of cases being category 1 and 2, and most are asymptomatic. When should we be considering to stop testing for these categories?
I think what you call it, the SOPs for that are already in place, right? You would have had your first test that told you were positive. And then if you're category 1 and 2, I think you are home quarantine or at the very least or in some quarantine center. And then you have an exit swab, 10th day, or probably with the Delta variant, you may want to go longer, maybe 14th day or so at least. And if your exit swab is negative, and then you are declared recovered, right? So I think that mechanism for determining whether a person is recovered and released from quarantine is already in place, and I think will continue. Whether we need to tweak it to be longer in light of the Delta surge with higher viral load numbers in an individual who's infected, probably needs to be looked at.
I would like to allow one question from the audience. If they have anybody would like to ask personally any questions, we'll allow one more question because we have, for the interest of the time. Okay, if not, let's talk to you. Okay, as I saw a raise of hand earlier. Never mind. Will Delta become like a common cold since most of our level 1 and 2?
Well, it may well be, but we have to remember, unlike the common cold, the 2 alpha and the 2 beta coronaviruses that are prevalent, there have never been any vaccines against them, right? All we rely upon is our own natural protection when we get the common cold. But we have seen that itself doesn't confer adequate protection at a long period of time because we get the cold almost every other year. But with SARS-CoV-2, very likely it will go the way of a common cold, or it may go the way of SARS or MERS. We don't see any SARS or MERS at the moment, and they are more closely related to SARS-CoV-2 than the common cold virus. But what we have now are vaccines, right? So we do not know yet how vaccines are going to impact us in the future, right? If we move ahead with the boosters, and increasingly that looks more likely than not, how long will this protect us? And if we can achieve very high vaccination levels in terms of percentage for the global population, whether there will be need, if we see a decline in neutralizing levels over the years, whether there will need to have booster shots like the flu. So those are things that we'll continue to monitor, I think, globally, and we will continue to together see how that will impact us in the future. Yeah. But certainly with vaccines, you know,
yes,
there's hope.
Definitely, indeed. Prof. Edmund wants to know your take on whether the controversial idea of the SARS-CoV being lab manufactured.
Well, the lack of any clarity in terms of data makes that very difficult to give an opinion on. So I don't know, right? That's something that we will leave to the people who are interested to investigate. I mean, there are proponents and non on either side of this with their own sets of evidences. But at the end of the day, I think what's important, it's already here, it's already a pandemic, it's already affecting us. How do we proceed? How do we control this? And how do we put in place, not just whether it's a result of a lab contamination or not, but increasingly, how do we put in place measures for labs that work with infectious diseases, particularly from a zoonotic nature and things like that? How do we increase awareness and level of protectiveness in these labs to prevent an occurrence of a virus escaping and causing things like this, or potential to cause things like this? Yeah.
Thank you very much, Prof. David. I think we have reached to the end of this session. Fred, we can't take any more questions at the moment. If any questions, I think we can directly email to Prof. David for further clarification on this. Thank you so much, everyone, for your participation, your patience, and staying with us through all this your lunch hour and listening to such an interesting talk by Prof. David for such amazing insight to coronavirus COVID-19. A reminder to all the participants, please click on the link provided in the chat, or the later we'll give you at the end of the session. And maybe the secretary can flash the QR codes for the MMC points and attendance. With that, I thank you very much, everyone. Have a good day. Assalamu alaikum.